right, welcome everybody. If you're in the room and if you're online listening to this section, you picked the right one. This is going to be some of the CBRN vaccine, antivirals, and antibacterials programs. It's going to be the most exciting breakout session that we're going to have throughout all of Industry Day. And I won't belabor the point here, but just thank you all for participating in Industry Day. Excited to have some of the people in the room and at least a lot more online at this sort of mix of in-person and virtual environment here, and hopefully we'll all be in person next year. But without further ado, turn over first to Karen Martins from the Antivirals and Antitoxins branch.
Let's see. All right, thank you. Does this advance? All right. And there we go. All right. Well, thank you so much, Dan, for the introduction. And my name is Karen Martins. I'm the branch chief for the Antivirals and Antitoxins branch. And I will get to my slides, I believe. Is there another deck that needs to be loaded? That's all I'm clicking through. Okay, there we go. Sorry. All right, great. Thank you so much. I will dive in here. So it's great to be here in person, and thank you to everyone for coming to this session and for those online. And I'm apparently technologically, it's not advancing. Okay, there we go. Maybe I need a stronger thumb. All right, apologies for that. So again, thank you all for coming. So just to give a little bit of background, so the Antivirals and Antitoxins branch targets really your traditional biodefense threats, filoviruses, smallpox, botulinum neurotoxin, and the anthrax toxins. Today I'll be focusing mainly on the filovirus, smallpox, and botulinum neurotoxin areas of our portfolio. So the anthrax space, we do have 3 approved products, antitoxins, Anthracil, Anthem, Raxibacumab. The botulinum neurotoxin space we'll talk about a little bit as we're interested in opening up our scope there a little bit and moving into host-directed therapeutics and small molecules. The smallpox space, well, certainly some of the products in our portfolio may have been on the news about the monkeypox response, and looking forward to talking to you a little bit about that. And the filovirus area is really the most active area of our portfolio right now, where we have 2 approved products for the Ebola virus indication and are working on Marburg and Sudan virus as well. Before diving into the portfolio, I want to flag an approval this year for the intravenous formulation of TPOXX. This is the result of a tremendous amount of work from the team at SIGA, as well as our PCT and project officers and the AVAT team. And the relevance of this obviously was quite relevant in light of the monkeypox response, which we'll talk about a little bit. But while the capsule formulation of TPOXX was approved for patients down to 13 kilograms in size, this IV formulation made the product accessible for smaller patients down to 3 kilograms in size. So really expanded our ability to treat pediatric patients and get that drug to patients who need it. So we're going to start off talking about our filovirus portfolio. As many of you in the field are aware, there are 2 products that are licensed for Ebola virus. So these are the monoclonals, Inmazeb and Ebanga. And this was the work of both the companies, Regeneron and Ridgeback, and now Emergent, and tremendous efforts through the PALM study in DRC in 2018 to demonstrate efficacy of those 2 products. We have active products in the portfolio for Marburg and Sudan virus. So for Marburg virus, we're working with MAP Biopharmaceutical on a monoclonal antibody product, MVP091. This is past phase 1, and so we're moving forward with process validation and moving that product towards licensure. For the Sudan virus indication, we've had a very active couple of months. So this is a 2 antibody cocktail developed also by MAP Biopharmaceutical, also past phase 1, and we had a small amount of GMP material that thankfully we've been able to leverage for the current outbreak response in Uganda, which is a Sudan virus outbreak that's going on right now. We're working very closely with interagency partners and also with WHO and the Ministry of Health in Uganda to make this product accessible in the near term through compassionate use, but obviously focusing on that randomized clinical trial and working with WHO to develop a protocol that can be implemented. So hopefully there'll be more to come on that in the news in the next week or 2. In speaking in terms of filoviruses, we've really had a paradigm shift, I think, in the last 5 to 10 years as we've learned more about these viruses and the long-term consequences of infection. Just in the last 2 years, we've seen the first Marburg virus outbreaks in West Africa, both in Guinea and Ghana. Previously, while Marburg virus had been detected in bat species, we hadn't actually seen human infection in West Africa. So again, potentially a signal that we're seeing either better detection, better surveillance possibilities, or also a new zoonotic incidence of these and zoonotic crossover into humans. We've learned a lot about long-term morbidity and mortality. So certainly coming out of the West Africa outbreak and the DRC outbreak in 2018, 2020, we learned that what we had seen as signals for long-term morbidity really is relatively frequent, that we do see these long-term sequelae associated with infection, and this has become an area that we're very interested in, finding therapeutics that can help treat the patients and treat this long-term morbidity associated with infection in survivors. And then there's also the issue of recrudescence. So I believe it was 5 of the outbreaks in 2021 were actually associated with recrudescent infection from survivors who had been cured, quote unquote, of Ebola virus months or even years prior. And so we do know that there's some sort of sustained infection that can resurge and cause a new outbreak or new infection in the patient. So while we're certainly focused right now, we have products in the portfolio for Marburg and Sudan, I do want to make the point that to BARDA, the Ebola virus issue is not solved. We're actively looking for therapeutics that can help treat severe infection, those patients with really low CT, high viremia, where the monoclonal antibody products that are approved are still not able to completely resolve the infection. We're still seeing about a 60% case fatality rate in those patients. Definitely interested in products that can treat persistent infection if that is what's causing this recrudescence, and that could be used potentially in combination with the monoclonal antibody products to improve outcomes. Oops, I went too far. All right, there we go. In the absence of outbreaks, so we just want to highlight some of the collaborations we have with the non-clinical group and vaccines group in developing animal models for filoviruses. In the absence of clinical data, we're of course pursuing the FDA's animal rule for licensure for products for Marburg virus and Sudan virus. While many who work in the clinical environment might think animal rule might be a shortcut or an easier way to get a product license, I would argue it's actually the much harder pathway. We need well-characterized models, ideally qualified models, qualified or validated assays, and certainly as we're thinking about emerging infectious disease pathogens that we haven't identified yet that we would like medical countermeasures for, it's very difficult to establish those models in a high-quality capacity with good laboratory practices and be able to run those pivotal studies to achieve licensure through animal rule. So this is a very active area of our portfolio, working with a non-clinical group. Also, I want to flag, certainly for those in the audience who are working in the filovirus space, the current non-human primate shortage is a real challenge. Many of our studies that lead into not just proof of concept, but also those pivotal studies would be in non-human primates. So we are working with UCAMRID to evaluate the ferret model as another potential avenue to look at therapeutics. But really at this point, the gold standard is that primate model, and I think it's something the community is thinking about of what we can do to solve this issue. So to sum up in terms of our approach for Ebola virus and also the other filoviruses, we're constantly assessing the products that we have in the portfolio. Are there ways to evaluate further the dosing, the frequency of dosing, how they're administered in the field? We are very actively interested in new products in this space, and so if you have a product that should be considered, please feel free to reach out to me. My information is both in the broad area announcement and also at the end of the slides. But we're also looking at ConOps. So we're interested in lyophilized products, reducing the cold chain, making these products accessible and easily administered in the field. And the long-term goal here, of course, is reducing that morbidity and mortality we see in survivors, improving outcomes, and also reducing this recrudescent infection. So I'm going to quickly touch on botulinum neurotoxin just in the interest of time, but we do want to flag that this is an area of interest for the branch. So as some of you may be aware, we do have a licensed product, HBAT, that's for the treatment of intoxication with the 7 serotypes of bot neurotoxin. We're also supporting an investigative monoclonal antibody approach with MapBio. We're having a lot of conversations about how do we address this requirement, this gap, in terms of treatment of patients who are already exposed to the bot neurotoxin, who already have symptoms. So we're actively interested, again, in sort of next generation candidates, small molecules that may be able to impact the toxins and neutralize them, but also host-directed therapeutics. So what are interventions that we can bring to bear to actually help treat the symptoms associated with intoxication? So moving over to smallpox therapeutics. So this is a space where we do have 2 licensed products, TPOXX and TEMBEXA. And as I mentioned earlier, we do have 2 formulations of TPOXX as well as 2 formulations of Tembexa, both of which are currently in the stockpile and available for use for patients as needed. As a risk mitigation, particularly for immunocompromised patients or for high-risk patients, we are working with BioFactura for monoclonal antibody cocktail. This is looking very promising in preclinical work, and so we're excited to move that to licensure as well. But the smallpox area has certainly been in the news recently in terms of the monkeypox response. And so I want to dive a little bit into our efforts with SIGA on the TPOXX product. So this is a product that is approved for the smallpox indication. And as those of you here I think are familiar that BARDA's funding is driven by those material threat determinations and threats that are identified as high priority for national security. But what I think is a fantastic lesson learned here is that this is a product that has efficacy in animal models excuse me, against multiple orthopox viruses. And so what, excuse me, still fighting off a cold. So what we were able to do, that having this product available and licensed for smallpox basically gave us a leg up. So this is a product that had validated manufacturing processes, we had clinical data, we had safety data, we had tox data, and so we were able to pivot to test and evaluate this product for monkeypox, and also to allow access to the product under appropriate regulatory mechanisms for monkeypox patients. And where we see the value of this is really an example of the type of product that has that broad spectrum antiviral activity against orthopox viruses, and a demonstration that we can advance it for one indication and then be able to pivot and use it for other indications as appropriate. So I want to highlight in just the last few minutes here some new efforts within the team. So this is Reboot, and this is a new effort that's a partnership with Drive and a new area of interest in the EZBAA. The goal here is that we recognize that developing products, particularly for filoviruses, can be a real challenge. There isn't a commercial market. It's very difficult to get access to BSL-4 labs to test and evaluate candidate products. And so the goal here is to work with companies that have late-stage products past phase 1, potentially licensed for another indication, that might have a mechanism of action that would lend themselves to efficacy in the filovirus space. The goal here is with the funding through this EZBAA mechanism to help support proof of concept studies, help fill potential gaps in the portfolio for advancing the product, so that we can consider whether this might be a small molecule or product that we could consider for filovirus support down the road. So please please check out the EZBAA put out by Drive. Happy to send links to anyone that's interested. We're very interested in hearing from you about products that may fit in this space. And finally, I want to talk about threat agnostic therapeutics. And so there's a lot of talk about this right now of when we get the next emerging pathogen, the next pandemic, the next outbreak, what can we have on the shelf to help treat those patients without knowing exactly what's going to cause it? This is an area that our chem and our rad-nuc branches have been working in for years, because really the approach for those classes of threats is to treat the patient, treat the symptom, and help to ameliorate the injury to the patient. So we're starting to think more about this in the biospace. Are there common themes, common symptomologies, disease states that patients present, particularly with hemorrhagic fevers, for example? Are there targets there that we can identify to develop host-directed therapeutics to help treat the patient, likely in combination with direct-acting antivirals? And so the idea would not necessarily to be the 100% cure, but to have something that can help reduce morbidity and mortality while we work on those antivirals and potentially monoclonal antibodies. We're interested in this space for filoviruses, also for bot neurotoxin, as I mentioned earlier. So again, products that would help reduce the symptoms associated with exposure and help the patients improve outcomes. So in summary, really where AVOD is looking going forward is this broad spectrum space, products like TPOXX that can be leveraged for other indications as needed, products that have that broad-acting antiviral activity that we can evaluate and determine an indication that makes sense to pursue the product, that we can test clinically, but that we also have preclinical data to support efficacy in other species or other virus families. We're working closely across BARDA in the host-directed space. Drive and IEID, other partners within BARDA and outside have a lot of interest in this space. And so we're coordinating efforts and expertise to understand what are those symptoms and syndromes that we can target and develop host-directed products that will help improve outcomes. We're also very interested in, of course, working with NIAID, with DoD, with other partners who are really supporting the same principles as BARDA's strategic plan of broad-spectrum antivirals, products that can have us prepared for the unknown. Certainly welcome any questions. I think we'll do those at the end, but that's it for me. If anybody has any discussion points, we do have the session this afternoon at 4, and I'll be on tomorrow at 4. Happy to have one-on-one meetings and talk further then. Thanks so much.
Great, thanks Karen for the overview of some of the recent successes from the antivirals and antitoxins program and where you're taking the program in the future. I have seen both Eric and Jessica exercising their thumbs, so they'll be better suited to work the clicker. And with that, I'll turn it over to Eric Stavali to talk about the vaccine program.
All right. This one? Okay, thank you for joining. My name is Eric Stavali with the vaccines program here at CBRN. So I'm going to talk about a few things here. My thumb is working. What we've been up to for the past few years, what our successes are, where we want to be soon, and what our long-term goals are. So, as in 2022, we're still seeing cases of COVID. There are question marks that are being raised with the new variants that are emerging. We've seen outbreak of monkeypox. We've seen outbreak of Marburg in Ghana, and we're actively seeing cases of Sudan Ebola virus in Uganda. So we have, as you know, we have vaccine that is licensed for the Zaire Ebola virus. Unfortunately, the glycoproteins between Sudan and Zaire Ebola virus are not similar enough to where we can use the vaccine for both strains. So we are actively, we're moving forward rapidly with our partners, IAVI and Sabin, in manufacturing and production of the vaccines that we have in our platform, in our pipeline to address that threat. So some of our recent successes in the last few years are licensure of multiple vaccines. So we had Biothrax in 2015, we've had Ervebo, and we've also had Jynneos for smallpox. I think, worth noting is, give me 1 second.
Just quick technical difficulty. Give me 1 second here. And while I was going to say, while we're waiting, maybe a couple quick questions for Karen. I'm going to take one real quick, and then maybe we'll see if there's any in the room here. But for your programs that are more mature, especially in the anthrax, smallpox, and botulinum toxin space, do you see a need for additional next-generation candidates, or are we at the stage where we can say mission accomplished?
Yeah, it's a tough question. I never want to say never. So certainly within the anthrax space, right now our requirement is, can you hear me with my, right now our requirement is for antibodies against the toxins. And so for antitoxins, we've really, in a way, checked that box. We have 3 candidates that are approved and highly efficacious. I think we're always interested in innovation. So if there is a new approach, a small molecule approach, something that, maybe a little bit outside the box, please reach out. We're happy to have those conversations and hear more about it. With smallpox, I'd really say along the same theme. We have 2 antivirals. They have complementary mechanisms of action. There's always, of course, the possibility that we see a situation where neither of them works, but these are DNA viruses, so we have some confidence that between those 2 we'll have good coverage, in particular with the antibody product in the pipeline. So I think BOT of those 3 spaces is the area that we're really interested in new and innovative approaches. We are already pursuing a monoclonal antibody approach. It is extremely challenging to develop a product that's efficacious against all 7 serotypes, which is the requirement. And so if somebody has a unicorn product there that you want to come talk to us about, we'd be happy to hear about it. But I think in BOT, where BARDA has not focused previously, but where we're looking to pivot is treating the patients, and so treating the symptoms of intoxication. And that's a space that we're certainly looking for partners.
And then maybe one more quick question. We heard Gary talk about the American Pandemic Preparedness Plan this morning, kind of focused on the vaccine goals of having vaccines within 100 days. Are there similar goals on the treatment side of the house?
Yeah, I won't put as much of a timeline on it, but I think the focus, whereas vaccines with the mRNA platform, there's a lot of focus on the platform technologies and that this is something that can pivot. Therapeutics, I would argue it's not, I won't say it's simple because you're vaccines, but I would say there are additional challenges in that you have to understand what the target epitopes are, you have to understand the host response if you're going to go with monoclonal antibodies. And so I think what we're really focusing on both across AP3, across the BARDA strategic plan, the National Biodefense Policy is broad spectrum antivirals to the extent that they're feasible, balancing that safety and efficacy. I think one of the challenges with small molecule antivirals is they tend to be a little bit, need to be given very early in infection, maybe a little bit less effective than monoclonal antibodies, as we've seen with viral hemorrhagic fevers at least. But there's absolutely space for something on the shelf that we can treat the patient when they need it while we start evaluating what maybe your gold standard or your direct species-specific product might be. So in terms of aligning with those strategies, that's really our focus area is the broad spectrum host-directed countermeasures. And then I'll throw in there for monoclonal antibodies, getting that cost of goods down. So we've been focused on CHO cell line products. They're extremely expensive. When we start talking about LMIC and the need to actually get these products out into the field, they are a challenge. And so new ways to approach monoclonal antibody development that we can get through the regulatory path and actually get approved is a big interest also.
All right, we will call in a pinch hitter for the vaccine program. So where were we? So in terms of success stories for vaccines and how well prepared we are for biological threats in general, we're reasonably well prepared for what I'll call the historical threats. So smallpox, anthrax, Ebola, we have licensed products. But we can do better, and we will for the next generation threats like Marburg and Sudan. So what we're doing is applying along the lines of the American Pandemic Preparedness Plan, similar concepts in terms of having licensed products for a prototype in that family. So we'll use Ebola as the example. So we have a licensed vaccine for Ebola Zaire. Unfortunately, as Eric was saying, the glycoproteins are different enough that that vaccine will not protect against Sudan Ebola virus. It won't protect against Marburg virus. So what we're doing is taking the same platforms, inserting the right glycoproteins for additional monovalent products for Marburg and Sudan. Hopefully now I can't figure out. Oh, I spoke too soon about practicing the. There we go. All right, all that said, we need to take Marburg and Sudan off the table as we prepare to look towards the next threats that we'll have to hit, other viral threats that have potential to cause pandemics in the future. So for Marburg and Sudan, and what we've talked about internally, and we've started to communicate this more with our external audiences, what do we actually mean by preparedness? So at the end of the day, we need licensed products. We need vaccines that are licensed by the FDA, that have emergency use listing by the WHO, so they can be used in country when needed. Because at the end of the day, for Marburg and Sudan, similar to Ebola, we need to stop these outbreaks where they're happening so they don't come back to the shores in the US. And what we're doing with Marburg and Sudan is exactly that right now. So we have vaccine candidates that look promising in terms of the non-clinical studies we've run so far. We have a candidate with phase 1 data that potentially would be poised to move into ring vaccination trials in Uganda in response to the current outbreak of Sudan Ebola virus if we have enough clinical trial material, which we don't at the moment. So what we really need to get to is a point where we have clinical data from a portfolio of vaccine candidates, along with enough clinical trial material that we can actually make a difference in an outbreak response. And in general, what we think that right ballpark material is 10,000 plus doses. The response scenarios have been pretty consistent since the West Africa outbreak from 2014 to 2016. With every index case we see, ballpark estimate, 100 doses of vaccines are needed. So 10,000 doses of a vaccine that might allow us to respond to an outbreak of 100 plus cases of Ebola, whether it's Sudan or Zaire or Marburg virus. And then we talk about traditional licensure versus the animal rule pathway for licensure. And for that intermediate level of preparedness in terms of clinical trial material and clinical data, that pathway is pretty similar. You can prioritize some of the different activities differently depending on whether or not you're thinking animal rule versus traditional pathway. But at the end of the day, to get to that stage of phase 2 clinical data and clinical trial material, the pathways run in parallel. The difference is when we start to get beyond that, and whether or not we see opportunities to demonstrate clinical efficacy in the field, or if we actually do need to pursue the FDA animal rule. And Karen spoke a little bit about this, but animal rule isn't necessarily an expedited pathway to licensure by any stretch of the imagination, because there are a lot of safeguards in there to make sure that the animal models they do recapitulate the disease, and importantly, they're predictive of what we would expect to see in vaccines or therapeutics. So the difference in the pathways really come down to what needs to be done post-phase 2, whether it's a phase 3 efficacy trial or a pivotal non-clinical efficacy study done in animals. So along those lines, we are thinking about what the next threats are that we'll need to be concerned about. So internally, we've conducted a series of what we're calling endgame workshops among the BARDA subject matter experts and some of our NIH colleagues to at least think about what other emerging viral threats have potential to cause pandemics and which threats we might need to prioritize in terms of next generation vaccines, and if any of the platforms that we're investing in could also be applied to these threats. So we've hosted a series of internal meetings to talk about things like Nipah virus, Lassa virus, et cetera, and are sort of at the stage where we're really conducting market research in terms of what the vaccine candidates are in the pipeline and whether or not there are ways we can make more sustainable approaches to vaccines in the long term. And what I mean by that is historically we've been limited in terms of budget, in terms of hitting one threat at a time. So we had anthrax with a specific vaccine, a specific manufacturer, a specific manufacturing platform. We had smallpox next, different manufacturer, different platform. Then we moved on to Ebola. Again, different manufacturer, different platform. So at this stage, we have sustainability issues on the back end for all of these programs as we continue to be successful in getting products across the finish line. We're trying to resolve some of that by investing in platform technologies. And I say this, but I hate the word platform technologies because there isn't anything that the true platform is make-believe. So if anybody has a better term that I can start using in the future, please give me a shout. But the idea is that we want either, whether it's a viral vector or RNA-based vaccines, something where we can swap in and out antigens relatively easily and be able to move quickly from the discovery phase through phase 1 clinical trials and then advance things to licensure as either opportunities come up to show clinical efficacy in the field or we have actual clinical efficacy based on either seasonal outbreaks for things like Lassa or outbreaks or for things like Nipah. Karen, I apologize, I spoke too soon on the thumb. Okay. Now, for the future of vaccines, what we really want to do is resolve some of these issues around sustainability, not just in terms of moving to the next threats and deciding which vaccine platforms we're going to use to hit multiple threats, but also looking at different ways to resolve some of the issues around stockpiling. So again, at the end of the day, if we get a product to licensure, we're on the hook to continue manufacturing, to stockpile to make sure that we're able to respond. But what we want to do is look at technologies that might enable us to rapidly transition between products when needed, and then improve the cadence at which we can manufacture. And what I mean by that is, to the maximal extent possible, moving away from large centralized manufacturing if it's feasible. So thinking along the lines like RNA-based vaccines, where we're starting to move towards more chemical synthetic processes, as opposed to the large-scale biological manufacturing processes, whether it's in eggs, in cell lines that are going to have inherent biological variability, to see if we can move away from those large centralized processes and move towards a network of distributed manufacturing technologies where the pipe dream might be if you had something of a logistical footprint that you could field it at the CVS's and Walgreens of the world. Obviously, a lot of technical and regulatory hurdles between here and there, but there could be an intermediate where it's just a more geographically based distribution network of manufacturing at smaller scales. So if it's hundreds of locations across the world, we might not need to be able to produce enough vaccine to vaccinate 300 million people at every location within 100 days. So divide that by 100, if you get 3 million doses within 100 days, maybe that's more feasible, but that only works if a technology taken up by the larger vaccine industry too. It's got to have a warm base, whether it's flu vaccines, shingles vaccines, something where there is a continued use, and then we can turn it on whenever there is the next pandemic. And again, I need to shout out to the partners here. I think we all include these slides, but we obviously can't do any of this without you. So we're going to continue to look for new partnerships to invest in flexible technologies that are going to let us hit the next threats that we're concerned with and do it in a more sustainable manner. All of our partners have been great to help get these products across the finish line, and these relationships are just so critical to our ability to do anything on the preparedness front and our ability to respond to things like the monkeypox outbreak, the Sudan Ebola virus outbreak, the endless Zaire Ebola virus outbreaks over the past 7+ years now. So just thank you to all of our partners. And with that, I'm going to turn it over to Jessica.
Thanks, Dan. My name is Jessica Swenson, and I have the esteemed pleasure of talking to you today about CBRN's Antimicrobials program. We are the branch formerly known as Antibacterials as of today. If you read the BAA before you go to bed every night, you would have seen last night that we are now investing in antifungals for the first time. And we are, round of applause in the room for those of you who can't hear virtually. This has been a big endeavor by our team. We've worked with a lot of interagency partners, and we've really realized that the AMR space is a lot broader than just bacteria, and we're thrilled to be moving into the antifungal space. So I have a lot to tell you about that, if I can get the clicker to work. I think Dan did take the batteries out. Let's see. Okay. So usually when I present this slide, I start talking about bacteria that can be intentionally engineered to become weapons. And today I feel compelled to tell you about a patient instead. So about a week ago, someone that I know and love was hospitalized in the ICU giving birth to her first child. Should have been the happiest day of her life, and it ended up turning into a tragedy. Thank God we have modern medicine, because she was able to be sustained on life support for a week, and she was able to start breathing on her own a couple days ago. What happened after that? She spiked a fever. What happened after that? She was given antibiotics. And today she's one step closer to going home and meeting her daughter. Antibiotics are the underpinning of modern medicine. And we know that with any public health emergency, we're going to see more hospitalized patients and patients with prolonged hospital stays. And we need to be prepared to treat those patients, whether or not there's a rad nuke event, or there's a hurricane, or there's just an everyday occurrence of a postpartum tragedy. We also know that AMR exists every single day across the globe. In 2019, we saw more deaths from antibiotic-resistant bacteria than we did from HIV or malaria. That's astounding. We also know that bacteria can be weaponized, and this is where BARDA really came into this space. So today we're here to tell you a little bit about what we're doing in this space and how we're moving into antifungals to complement what we're doing in antibiotics. Oh, we're going backwards. Okay. So what's bolded on this page is our newest indication, fungal infections. But of course, we're really working across BARDA's strategic plan to make sure that we're able to create medical countermeasures for a whole host of threats. We work in biothreat pathogens. We also work in everyday AMR pathogens. Our priorities recently have shifted to be able to focus on things that are acquired in the hospital and also in the community. We're really looking at pneumonia and bloodstream infections as some of our targeted indications. And we're looking for things that can be easily delivered to these populations, maybe oral formulations, intramuscular, different things other than just IV. We're also looking to how we can serve special and underserved populations, pediatrics, as Karen was mentioning. And we're looking to see how we can start creating a portfolio that builds on the successes that we've had over the last 12 years and how we can keep moving the needle. Because we know that antibiotic resistance keeps developing faster than we're able to create new products. So this is a snapshot of our portfolio as of today. We have 55 active projects, and they span all the way from hit to lead, which is a very early preclinical stage, all the way through procurement. Our CARB-X program is one of our flagship programs, and we're so thrilled to be able to support antibiotics all the way from the earliest stages of development through the procurement. Our advanced research and development portfolio currently has 6 products in phase 3 clinical trials, and we're hopeful that those will be able to achieve licensure soon. This year, we've also initiated our second Project BioShield contract for ceftazine pendimetabactam to be able to be used as a potential treatment for melioidosis indications. And of course, none of this would be possible without our partners in development. At the bottom, you see just a host of some of the interagency partners that we work with, but really it's industry. It's all of you who are watching today who enable us to do this work. And as Karen and Dan mentioned, this is a partnership. We work with you. We work together to meet our common shared goals. Oops, no, too fast. Okay. So if you're new to BARDA and you've never met the antimicrobials program before, what are our needs? In the orange boxes, you see a little bit about some of the critical questions that we ask ourselves. And you see in the bottom some of those things that we're really looking for you to bring to us when you're coming to have a conversation. Do you have a novel approach? What does your development pathway look like? What is the FDA saying? And how can we get there together through a cost-sharing platform? And keep in mind those special populations, things like pediatrics, pregnant women, those who have kidney dialysis needs, those are all American population, and those are all part of our constituents. And we make sure that we're able to create products that fill all of those gaps and leave no one behind. Like this. Okay. In 2020, we've started bringing in some novel and non-traditional approaches. We currently have microbiome and phage in our antibiotics portfolio, and we're excited to keep bringing new and innovative technologies into this space. We know that there's potential for non-traditionals to circumvent resistance and to be able to have reduced side effects. So if you are working in this space, come talk to us. We're eager to hear what you have to work on. Antifungals. So later this week, we'll have some additional social media that will be coming out to really highlight what we're looking for in this space. But as I mentioned, the BARDA BAA has been updated to include a new sub-area of interest. It's number 3.2. And this sub-area of interest talks about what we're looking for in antifungal candidates. We're hoping to really prioritize things that are broad spectrum and that can overcome some of the challenges that we see with existing treatments. We're going to be working in the Candida species, also in the Aspergillus species, and we'll be considering rare molds as well. If you're a candidate in this space, we're looking for a technological readiness level of 6, and we are thrilled to be able to start a tech watch, if they can click one more time. Aha. If you have a product in this space, we want to talk with you. If you go to the BARDA BAA website, you'll see on the Tech Watch page that there is now an area where you can request to come in and speak with us about an antifungal. Give us a good description of what your product is, and we're happy to come and have a conversation with you. If you can do this before the end of the calendar year, it makes our lives that much easier because we're hoping to evaluate candidates early in the new calendar year and be able to start trying to hopefully make a an acquisition late in FY23. I want to round out the talk with just a couple of achievements from this year in FY22. So in the summer of this past year, we awarded a 10-year additional contract to the CARB-X program. I usually ask people who here knows of the CARB-X program, and everyone raises their hand, so I'm not going to ask, but CARB-X really is one of ASPR's flagship programs in the AMR space. It was started in 2016 by BARDA, NIAID, and the Wellcome Trust, and since then, we've grown to include also the UK and German governments, the Bill and Melinda Gates Foundation, and this year we announced an additional 10-year partnership for an additional $300 million on top of the original $200 million that we invested in CARB-X. CARB-X, if you remember the early pipeline slide, is the early stage part of our development pathway, and they really work across 3 different pillars that they call aligned by design. They are therapeutics, vaccines, and diagnostics, which come together to create a a whole portfolio working toward getting things through phase 1 clinical trials to be primed for BARDA or other late-stage funder. Oh, I do want to mention, CARB-X is recently announcing that they will be having new funding rounds. If you missed the funding round in October, they will be having additional funding rounds open in January and April of 2023. So check out the CARB-X website if you're interested. And lastly, as we get into Project BioShield. So Project BioShield on that timeline is the very end. This is where we get into procurement, and it's mostly for biothreats. So really, this is our health security preparedness posture. And it's also one of our avenues that we can help support companies through their post-marketing commitments and to domestically onshore products to be able to have antibiotics here made in the US. In 2019, we initiated our first Project BioShield contract for NuZyra through Paratech Pharmaceuticals. And just last month, we initiated our second Project BioShield contract for cefepime-taniborbactam with Venatorx. These are both to study biothreat pathogens and potentially get toward indications that will create a preparedness posture that enables us to make sure that we are able to treat weaponized bacteria. And with that, I just want to leave you with a decade of what we've been able to do. Our team is amazing. You hear this all the time, that the BARDA family is a BARDA family. And what we are able to do with our industry partners and with our interagency partners and our international partners is amazing. We've invested over $1.7 billion in the last 12 years. And together that's created over 120 different projects in a portfolio that's moved the needle for antimicrobial resistance. So I'd be remiss if I didn't thank the entire team of subject matter experts, federal staff, amazing BARDA leadership, and our partners as well. Thank you for all that you do. We're looking forward to working with you in the next coming fiscal year. Thank you.
All right, I think this session goes until 2:30, unless somebody, all right, Christy shaking her head, yes, that's a good sign. So there are a few questions that kind of overlap across the threat space on the biological side. So one that I was gonna throw out there for all three of us to answer is, how do we prioritize among threats, especially where there are not yet licensed countermeasures? And Jessica, maybe we'll start with you.
Yeah, it's the challenge, right? And I think in the antibacterials and antifungal space, this is where we really rely on our interagency partners, right? We rely on looking at what the global landscape is for antimicrobials. Where are we having gaps within our own portfolio? We've made a really concentrated effort over the last several years to really be strategic in what we're bringing into the portfolio, making sure that we have enough candidates to backfill things when we know that there will be attrition just for technical challenges alone. But also in the antibiotics and antifungal space, we have a very challenging commercial market. And those are all things that we look at when we holistically evaluate our portfolio. We are very lucky in that we have the CARB-X portfolio, which helps to feed some of those early preclinical things into the advanced research and development portfolio. And it allows us to see what's coming down the pipeline. And through that, we're able to prioritize and make sure that we have a full spectrum of things to be able to treat the most difficult to treat infections.
Yeah, thanks. So for antivirals and antitoxins, I think I'll look at this from two angles. So one is the threat space that we're currently working in, which again is filoviruses, smallpox, spot, and anthrax. So it's a little bit limited by what we have material threat determinations for and what we can, so to speak, fund at this point. So it's easy because Marburg and Sudan are the big gaps. We're certainly looking for products in that space, and then we're looking again for those products that span and have broad filovirus activity. We're putting a lot of thought right now with interagency partners, especially working with APP and the NIAID team, as well as with DoD and accelerated antibodies to think about what comes next. And so I think that's on everyone's mind as we're, quote unquote, coming out of the COVID-19 pandemic. What are the next viral threats or next bacterial threats that may be naturally occurring outbreaks, the cousin of the Lassa virus, the cousin of chikungunya, something that maybe we haven't identified yet, but has that potential to cross over from zoonotic species and become a problem for humans. And so we have groups, in addition to the endgame workshops that Dan's group's been doing, we've been working with CAV and APP to put together target product profiles, to think about balancing feasibility and also availability of products. So We've done an assessment of what candidates are out there based on literature searches and talking to companies. But we also need to think about feasibility of test and evaluation. So what are the targets that we can actually evaluate in a clinical trial? What are the manufacturability concerns about a given product class? And is it something that we can scale up and advance? So it really is a matrix decision of the morbidity and mortality associated with the threat, the likelihood of an outbreak, and then pragmatically what's out there that BARDA can help support at this point.
And I'll just echo all of that, because Karen and I share a lot of the same threat space, but for the near-term priorities, that's exactly it. It's the filoviruses that have not been knocked out yet for our programs. But on the vaccine side, again, we're really looking towards the next threats in terms of emerging viral threats that may be of concern, because one thing that'll be a little bit different is whether or not there are sort of clinical operation perspectives that work for the threat. So something like botulinum toxin, where an antitoxin may make a lot of sense. Unless we're going to mass vaccinate against botulinum toxin, having a stockpile of bot vaccines may not make a lot of sense. So that's something that we might not prioritize investments in. So it's really those emerging threats where we can intervene at sort of the outbreak stage before things become full-blown pandemics. And since you both spoke to it, kind of the next one that was kind of an overarching question was if and how we work together with the interagency. And we'll just take the reverse order here. But for the vaccine side, we work really closely with our DoD partners at the Defense Threat Reduction Agency, Joint Program Executive Office. Gary spoke a lot about the partnerships with the JPEO through the COVID programs, but that applies across the board for our programs. And then our partners at NIH and the Division of Microbiology and Infectious Diseases, they have a lot of earlier stage programs. And then a big piece of what we're doing for the next threats is working with partners like CEPI that have the bandwidth to move forward into things like LASA and NEPA, and make sure that we have a finger on the pulse of what that pipeline looks like. So if and when we do have to pivot to the next threat, we're ready to do so.
Yeah, and I would echo that. Again, we have similar space, but I'd also say it's useful working with partners at DoD, for example, that have complementary, but maybe slightly different requirements. So whereas DoD might focus on PEP or PrEP, we can work with them on products and make sure that we're advancing them in a way that is useful for both agencies and for both needs and requirements. But definitely also echo, CEPI is becoming a new partner on the therapeutics and the biologic side. It's great talking with them and making sure that we're being complementary and not contradictory also. That we're not funding the same programs, that we're aware of what's coming down the pipeline, that NIH, that DoD have an earlier development that might be something that BARDA can help support as it's kind of goes through that screening process and is a viable candidate. So it really is a partnership of ensuring we're not being redundant, and we are aware of what's out there. So we really encourage that interagency discussion.
Yeah, and we're very much aligned, working very closely with all of our USG interagency partners, but it's even broader than that for antimicrobials because AMR is such a global challenge. So we work very closely across the entire U.S. government for the CARB National Action Plan. We work with our TAPFAR partners internationally, and we have a whole host of for-profit, nonprofit, dilutive, non-dilutive partners that we work in this space because AMR is the next silent pandemic, and because there is so much global attention on this issue. So we actually have the ability to have a whole host of partners outside of the U.S. government as well that really help to complement what we're doing with our interagency partners in the USG.
And we will have time for a few more questions. So if there are any in the room, let's say jump up to that mic, because I'm assuming you'll need to do that to be heard online. So I'm going to ask one real quick while anybody gets the courage to step up to the mic. So Jessica, you mentioned TRL6 for the antifungals program. Can you get into a little bit of what that actually means, and is it related to the CARB-X? Can you invest in antifungals in CARB-X?
Great question. So little reminder, if you remember bringing up the slide, CARB-X comes through phase 1, and then typically our advanced research and development portfolio comes through phase 2 and later. So this is where that TRL of 6 would come in for antifungals. Currently, we don't have a space within the CARB-X portfolio to support antifungals, and the reason for that is because those products are really funded through NIAID's portfolio at this time. That being said, the CARB-X portfolio is a nimble partnership that's able to change scope should all of the funders agree that this is a space where we need to see additional funding or additional support. So there is that possibility, I would say, at some point in the future if it's needed. For now, NIAID, I think, is your best bet if you're an early stage antifungal company. But if you're phase 2 or later, we want to hear from you.
Robin?
I don't have a
Oh.
question, but I can help bring the mic.
Great, thank you. Any questions in
Does
the
have a
room?
question.
All right, if not, one more. This one's probably for me and Karen, maybe a little bit more than for Jessica, but with outbreaks occurring overseas, Karen, I'll start with you, but is there anything you look for in a partnership in terms of clinical capabilities for those types of outbreak responses in sort of resource-constrained settings?
Yeah, it's a great question, and certainly as we're thinking about expanding the portfolio and other threats that we'll work with, for the most part, a lot of these viruses that we're tracking as the next priority, those outbreaks occur overseas, and they may be sporadic and they may be unpredictable, and so understanding what our options are in terms of engagement for clinical studies and what our partners would be looking for or needing in terms of support, in terms of training and data management. Regulatory alignment is certainly a challenge because different countries have different regulatory authorities, different ethics committees, and understanding how we work together in a way that's mutually beneficial is going to be very important going forward. Currently, for the Sudan virus outbreak, we're working closely with WHO, who's serving as sort of a mediator role, which I think is very appropriate, and it helps broker those conversations with in-country partners. But I think this is an area that is going to become very active for BARDA of understanding what we can do here.
Yeah, I agree with all that. We would definitely see that as an added benefit for any program coming into the portfolio for exactly those reasons. Once we talk about clinical trials overseas, it just becomes a lot more complex because our end goal here is FDA licensed products, so there will be FDA engagement, but having multiple regulatory partners that will need to be engaged and to get sort of buy-off on clinical trial designs, et cetera, becomes way more complicated. So having that type of experience is definitely an added benefit to the program. We got one here.
Hi. Hi, thank you for the talk. My name is Amari Jones. I actually work with BARDA. And you actually just brought up a great point that I wanted to even piggyback off of further is you mentioned earlier about chemical synthesis as being the priority pathway for developing vaccines or new therapeutics. So when we're thinking about FDA approval, are there mechanisms to work with the FDA when these trials are progressing, given that some of these pathways or these platforms, as we don't like that word, are formed, and, this is something that is always a hiccup for companies where they have a potential pathway or platform that they want to do, but it's a huge headache trying to get approval from the FDA to get it through trials. Over.
Yeah, maybe I'll start with that. So there are ways to expedite things to a degree. The VSV platform, vesicular stomatitis virus, is a good example where it's been applied to multiple threats so far, at least in terms of getting things into the clinic. So there's only one licensed product that uses it, but there's, I'll say, a reasonable level of confidence that if you put in a glycoprotein for another virus, as long as you can manufacture it, it's going to perform similar in terms of a safety profile. So the threshold for moving into the clinic might be a little bit lower in terms of the number of sort of toxicology and biodistribution studies that need to be done, because with the first generation VSV-based products, there was a lot more work in terms of non-clinical toxicology because it is a live replicating virus. So there are situations like that where there can be expedited pathways, or at least in terms of a more streamlined pathway in terms of a non-clinical development portfolio. I don't know if there's anything you want to add from.
Yeah, I just wanted to add on the FDA thread, because I think that's something maybe I know I haven't talked about too much, and I think is very critical. So BARDA, and the way I think about it, plays sort of an interesting role in the juncture between the companies and the FDA and the regulatory authorities. And we're really agnostic, so we don't benefit from the products. I can't own stock in any of the products I'm supporting, so I don't directly benefit in any way from products getting approved, but we all benefit from the best product getting approved. And so where I think, you know, we have a role. We have great communications with FDA. We within our space, and so within the antivirals, we have good communications with CDER. But I think it's an area that as we start thinking about these new approaches and certainly the platform technologies or other approaches, you know, one of the risks for monoclonal antibodies, for example, where everyone's familiar with the CHO cell pathway. It's a little bit scary for a company to start dabbling into other technologies that don't have a proven regulatory path. And so I think that's somewhere that BARDA is certainly interested in and has a track record of doing, but we need to do more of going forward as we start thinking about new approaches. Let's talk to the regulatory authorities and have that conversation up front so that we know what the hurdles are and we can work with the companies to help streamline that as much as possible.
The only thing I'll add is that when companies have the pleasure of working with BARDA, it's not just money, right? It's the subject matter expertise of a whole team that supports your company and your asset to licensure. And we are so blessed to have the regulatory and quality affairs team that has a ton of FDA experience that really makes us ask those tough questions and helps come up with a development path. We work in the non-traditional space in antibacterials, right? And there are a whole bunch of unanswered questions there. And we help think about what are those critical things that we need to answer, what is the best development pathway, and how can we get there together as a partnership and make sure that we are creating the best product and getting it across the approval line.
Excellent, thank you. And yeah, just want to say that one more time, just because what Jessica said is absolutely true. Whenever there is a partnership with BARDA, it is a partnership and a lot of subject matter expertise. So hopefully it is a two-way street in terms of us benefiting from the technologies that you all are developing, and then us being able to provide not just funding support, but also that sort of technical and quasi-consulting support because of the expertise that is in-house. And there's, a lot of that day-to-day where there is no regular day-to-day, we've been in a response mode for, well, at least as long as I've been at BARDA. So unfortunately, I think that is going to be the situation for the foreseeable future. But with that, I just want to thank everybody for attending and listening in, and feel free to reach out with any questions.